that both reading frames of this INC4A gene evolve rapidly, but this agreement is not between the frames. The disagreement is between the fact, the crucial role of this gene as a tumor suppressor and its rapid evolution. We don't deny that this is a rapidly evolving gene. Indeed, as you can see in the table, in the figure 5.5, both reading frames evolve faster than genes counterparts in the both tumor suppressor pathways it participates in. So we do not deny that the gene is evolving faster. It even makes our point stronger, since it evolves so fast in many species. How it happened that there is no stop codon introduced in multiple species in any of the reading frames. It makes our case as for functionality of this gene and both reading frames. It makes it stronger. And as I said, discordance is between the high evolutionary rate, which we did notice for both reading frames, and a crucial function as a tumor suppressor.
How will I do this? My time, Mr. Chairman.
I would like to move to the next.
Okay, well, continue discussion.
Yes, are you satisfied with the answers so far? Clearly, some further discussion is possible, but for the sake of time and other opponents, I would like to move to the next opponent, Dr. Katerina Markova. She is Assistant Professor of Comparative Genomics and Bioinformatics from Penn State University, United States. Welcome, please.
Thank you, Mr. Chairman. Dear candidate, I have a few questions that I would like to address on the authority of the rector of the university and by virtue of my right. The first, I would like to congratulate you on this occasion. Your dissertation is a very interesting work and makes for a very entertaining read. And I also found your cover and the illustration very fortunate and very nice. So I would really like to see this allowed in the United States, such form of presentation of the dissertation. And unfortunately, we have it in a much more formal way, not this nice way. At any rate, the first question I have to the candidate concerns your work on the transitive techniques. You present the use of transitive techniques in two chapters, chapter 2 and chapter 3. In chapter 3, you are realigning the CFTR region, and you're finding much more homology by the use of this new method that you developed. And in your final chapter, in the summarizing chapter 6, you're mentioning that transitive techniques work very well with genomic regions which are not the subject of strong evolutionary constraints. My question is, have you tried applying this technique to other regions outside of CFTR?
Learned opponent, thank you for your nice words about my thesis, and thank you for the question. So, this 2 megabases of CFTR regions which we compared, and we applied these transitivity methods to derive more orthology, or to find more homology between different species. At the time we performed this research, it was the only data set, the only region which was available for 14 or so species, sequenced in multiple genomes, and that we actually could use this transitivity property on large scale. So at the time we didn't have any other data. But this is a very good point, and we would like to follow up the research and try to align the whole genomes, as more of them are available nowadays. So we could confirm the result of detecting 10% more homology in this region between human and mouse than is just detected by pairwise alignment. I think it's a promising result, and it would be nice to see it generalize it for the whole genome, as this method excels in detecting homology of fragments which are under weaker or no evolutionary pressure.